Movement is our means of interacting with the world. It's how we translate our intents and our desires into action. My name is Eiman Azim. I'm an assistant professor at the Salk Institute, and my lab investigates how the brain controls movement. I think one of the most impressive accomplishments of our motor system is our ability to use our limbs to reach for and manipulate objects. If you think about all the ways you use your limb to interact with the world in your daily life, whether it's buttoning your shirt, driving your car, typing an email, it becomes apparent how debilitating it would be to lose the ability to perform these types of dexterous movements. And despite the susceptibility of these types of movements to neurodegenerative disease and injury, we still know very little about how the nervous system actually controls these types of behaviors. So in my lab, we use powerful genetic tools in mice to dissect and tease apart neural circuits and try to figure out how each of them contributes to controlling these types of behaviors. Now, this goal demands sophisticated tools for dissecting neural circuits and similarly sophisticated tools to dissect and quantify behaviors. We try to take an interdisciplinary approach to tackling central problems in the neuroscience field by merging the big conceptual frameworks offered by systems neuroscience approaches with the strengths of a bottom-up molecular dissection of neural circuits. For example, one major issue that we're confronting is how do we actually identify all of the roles that a specific neural circuit plays in controlling behavior, given that it's very unlikely that any one circuit has evolved to control just a single behavior. If we want to make more meaningful progress in understanding the neural basis of behavior, what we need are more comprehensive, quantitative, and naturalistic tools to quantify these types of skilled movements in the mouse. To address these challenges, my lab is developing new ways to characterize behaviors, and we're combining this with genetic, anatomical, electrophysiological, and imaging technologies to try to figure out how mammals accomplish such an impressive repertoire of movements, and importantly, how injury or disease can have such a devastating impact on the motor system. This research will offer a novel way to explore fine motor control, providing a more standardized, comprehensive, and high-throughput approach for exploring the neural basis of behavior. And while developed in the mouse, these technologies in principle can be scaled to other animal models, providing a versatile cross-species resource that eventually could help lay the groundwork for a better understanding of human motor function and dysfunction. So I think, of course, as scientists, it's our job to envision a future, but I think it's equally important that along the way, as we're doing this, we remain open to deviations from the path. We make sure that we don't build any barriers that don't need to exist. We don't create any dogmas that are unnecessary. And I think it's this kind of open and fluid environment that creates the necessary ingredients for these world-changing, surprising discoveries. And I think the Salk Institute really provides an amazing environment, an amazing incubator for this evolution of ideas, this dissolution of barriers, so that all of us together as a community can bring science forward to that future that we haven't even yet envisioned.